Hello, class. Well, let's see how this works. This will be part one of male reproduction. On the first slide, the overall function. For the male, as you can see there, the primary function is the production of the gametes, the male sex cells. And another function tied into that will be obviously the transfer of these gametes towards the female. On the female side, I know this is kind of out of place, but on the female side is to produce her own gametes through the process of oogenesis, and we'll talk about that one next week. The female reproductive system is also designed to receive male gametes, and this is a perfect example of internal fertilization. But also it is to provide a place, as I said, for fertilization to take place, but also implantation and future development of the developing embryo. So as far as the male is concerned, we can break this down into a number of subcategories. There are the primary sex organs, and this would be the paired testes, both the right and the left. We have the secondary sex organs, which are a series of ducts or tubes. And this would be the seminiferous tubules, and we'll talk about those today. The epididymis, the ductus deferens, the ejaculatory duct, and then the urethra. The third component of the male reproductive structures will be the various glands, and these will be the seminal vesicles, the prostate gland, and then the bulbourethral gland. To begin with the primary sex organs of the testes, developmentally, these structures developed internally on the posterior abdominal wall, so within the abdominal cavity. And then around about the seventh month of embryonic development, they began their descent through the inguinal canal until their final position was outside of the body cavity contained within the scrotum. And the primary reason behind that, spermatogenesis, the formation of the sperm cells, can't occur at body temps of 98.6 degrees. It requires temperatures 2 to 4 degrees cooler. So that's why we see these structures located on the outside of the body. It is interesting to note that not all mammalian species do we see this phenomena take place. Some indeed are actually, when I say the testes are actually retained inside the body cavity, but that's a topic for another discussion. But what's kind of interesting, in passing through this inguinal canal, the testes are going through several layers of tissue. And I know that we really haven't had a whole lot of time to talk about this in lab, but if you can think for a minute about the abdominal musculature, where you've got the external oblique, the internal oblique, the transverse abdominis, and of course the connective tissue or the fascia associated with that, but also you have the parietal peritoneum lining the abdominal cavity. So in order, as these testes again kind of make their way through, passing through this inguinal canal, they carry with them some of these other structures, and this becomes what's known as the spermatic cord. So you can see a couple of things, and you'll see an image on the next slide. I'll just kind of read this, but the external spermatic fascia, which can be traced back to the external oblique musculature, but specifically the fascia of the external oblique. The cremaster muscle, which is indeed a continuation or a remnant, I probably should say a remnant of the internal oblique. The internal spermatic fascia is the fascia which is associated with the transverse abdominis, one of the deep abdominal muscles. And then a structure called the tunica vaginalis, which is actually the parietal peritoneum, which is basically has been brought outside of the body. So these different structures, along with the testicular arteries, the papiniform plexus, some nerves, and another tube known as the ductus deferens, these all become incorporated into what's known as a spermatic cord. And here's just kind of an illustration looking at this structure here. And I'll post this, and you can go ahead and kind of spend some time. But if you look at the colors, beginning from outside going in, external oblique is an orange, gray would be the internal oblique, and that kind of that burnt orange, or that kind of reddish color, that would be the transverse abdominis there. But you can see the structures which eventually become incorporated into this spermatic cord. And here's just another illustration looking at the same thing right here. There's a structure you'll see here called the gubernaculum, and this actually attaches to the testes when it was in the abdominal cavity, and this piece of tissue actually kind of pulls and directs the descent of the testes into the final resting place there inside the scrotum outside of the body cavity. So as we said, we have the primary sex organs are the paratestes. In this particular cross section, I want you to look at what's called the seminiferous tubules. And it's within the seminiferous tubules that that spermatogenesis is taking place. Last week we had an exercise that was talking about mitosis and meiosis. So it's within here, the seminiferous tubules, that meiosis is taking place, and where the sex cells are going through those meiosis 1, meiosis 2, and the end result was making 4 haploid sex cells. The tubules themselves in cross-section, I'll show you that in a minute. The bulk of the tube, what are known as Sertoli cells, resemble simple columnar epithelial cells. They are an epithelial cell, and we've talked about that before, how epithelium will form ducts or tubes. But in addition to that, we have some stem cells known as spermatogonium that are also located there. And it's the spermatogonium that will give rise to the primary spermatocyte, which will then experience meiosis 1, going to the secondary spermatocyte, which then goes through meiosis 2, forming the 4 haploid spermatids. So this is looking at the seminiferous tubule in cross section, just one of these tubules, and you can see kind of in the blue color. There's the Sertoli cells, again forming the bulk of the tube, and kind of sandwiched in between would be the spermatogonium. These are the future spermatids. And then of course you can see down towards the center would be the lumen of the seminiferous tubule, where the mature spermatids would then enter and then eventually make their way to a temporary storage a receptacle known as the epididymis. Also in this illustration, I want you to look at there on the outside of the seminiferous tubule, you'll see a cluster of cells kind of in dark blue known as the cells of Leydig, or sometimes referred to as the interstitial cells. And these cells become very important because this is where the male sex hormone testosterone is going to be manufactured and then released. So spermatogenesis, okay, is a process which takes place within the seminiferous tubules here. It begins at puberty under the influence of follicle-stimulating hormone. Again, one of the hormones that's released from the anterior pituitary gland. And unlike females, spermatogenesis is a continual process. So again, from puberty until the death of the individual. This is an ongoing process, and what makes that possible is those spermatogonial cells, when they divide through mitosis, one cell stays behind. One of those new daughter cells stays behind to keep the process going. The other daughter cell will then go down this meiotic pathway, eventually giving rise to those 4 haploid cells. So that's why this process is able to continue. And here's just kind of a different illustration showing that. So again, if you can see the spermatogonium, okay, up towards the top, going through mitosis, and as I said, one of those cells will stay back and give rise to future spermatogonium cells. The other will go down and become that primary spermatocyte, going down that meiotic pathway, eventually giving rise to the spermatozoa, as you can see. And here's just another illustration, which I think does a better job. So you can see up at the top where it says spermatogonium, goes through mitosis, and it says daughter cell A again stays behind. Daughter cell B again will go down the path to the primary spermatocyte, secondary spermatocyte, and eventually the spermatotids. What's nice about that illustration as well is you can see the events of meiosis 1 and meiosis 2, again, where they're taking place. Remember, in meiosis 1, specifically prophase 1, is where we see the crossover event taking place between the non-sister chromatids of the replicated homologous pairs. And remember, the key thing about that is that created the variability or the shuffling of the genes, which explains why the human race, again, we're not, in a sense, clones of one particular phenotype. So that crossover event becomes very important. As I said, it does create for what's known as or called genetic diversity, genetic variability. Okay, now those Sertoli cells that are an epithelium, one of the important things that they'll do is they form a series of tight junctions, what's known as a blood-testis barrier. And in doing that, they prevent host antibodies from gaining access and destroying those developing spermatotids. Now remember, these cells are becoming haploid, so they do not have the same full genetic complement as a normal host cell. So as far as the body is concerned, this is a foreign entity. So this is why we see these tight junctions taking place or formed between the neighboring Sertoli cells and forming this blood-testis barrier. In addition to that, these cells need to be kept alive. Again, these spermatocytes, and they will basically provide the nutrients necessary to keep those individual cells alive. They will also assist in moving, again, these stem cells, again, down towards the lumen of the tubule. There's a substance which is called testicular fluid, which helps, again, move those, again, spermatozoa down and then eventually up towards the epididymis. Now keep in mind that these haploid spermatotids, or what you'll see this term spermatozoa, they're not motile yet. Again, even though they do possess a tail, they don't have the ability to locomote, again, on their own. So once they have gone through this developmental aspect, they still have to go through a series of modifications till they do indeed become fully functional cells. And here's just another view of looking at the Sertoli cells, and you can see in this illustration the tight junctions This also does a very good job of, again, showing you primary spermatocyte, secondary spermatocyte, and then the 4 haploid spermatotids that are being formed. And keep in mind that it's the testicular fluid, again, which is also kind of helping in this process, as I said, moving down the lumen of the tubule and eventually taking up residence within the epididymis. And here's just another view. And then here's another view too, a micrograph. And as we said, they're located between these cells of lydate, between the individual seminiferous tubules. And they, under the influence of luteinizing hormone, also released from the antipituitary gland, will begin the synthesis of the male sex hormone testosterone. And testosterone, not only will it, again, promote further development of the testes and things like the prostate gland, but it's also responsible for what we would refer to as secondary sexual characteristics. And such things like, basically, when you look at a male and a female, again, what are those basic differences? Increase in bone mass, increase in muscle mass, body hair distribution. So I'm thinking more and more in terms of facial hair, those types of things. A deepening of the voice as a result of thickening of the vocal cords. And of course, the distribution of body fat, where you see it located on females in particular areas. Again, normally you don't see that